Swayam Prabha. Digital India. Educated India.
Hello learners, today we are going to learn about forces of evolution. This is the sixth module under the paper Evolutionary Biology. In the previous lecture, we discussed forces of evolution in qualitative perspectives. Now we are going to talk about quantitative studies with respect to forces of evolution. In other words, we are going to talk about population genetics or the Hardyweinberg law. I am Dr. Sudhir Verma, working as Assistant Professor in Department of Zoology, Deen Dayal Upadhyay College, University of Delhi. In today's lecture, we are going to learn about Hardyweinberg equilibrium. We are going to discuss what this law or equilibrium states. What are the assumptions that it requires to be applicable on a population? Without these assumptions, this law is not applicable on a particular population. We will derive the mathematical expression for Hardyweinberg equilibrium. Besides this, we are going to discuss about various evolutionary forces that can upset this Hardyweinberg equilibrium. Particularly, we will restrict our discussion to four major evolutionary forces, which can be the natural selection, the genetic drift, mutation, and migration. So, we will see mathematically how these forces can change or modulate the Hardyweinberg equilibrium. Population genetics is the study of genetic makeup of groups of individuals and how a group's genetic composition changes with time. In order to explore the evolutionary processes that shape the genetic variation, we must understand genetic structure of a population, and this structure has to be understood in terms of frequencies of genotypes and the alleles, which in other words means the allelic frequencies and the genotypic frequencies. So before going ahead with understanding population genetics or Hardyweinberg equilibrium, we need to talk about allelic frequencies and the genotypic frequencies and how do we calculate these two. The genotypic frequency, as the name suggests, is the frequency of a particular genotype within a population. So it can be calculated by adding up the number of individuals possessing a particular kind of genotype divided by the total number of individuals in the sample or the population. It gives us the expression for the frequency of that particular genotype within that specified population. For a locus with three genotypes, let's say the dominant homozygotes, heterozygotes, and the recessive homozygotes, that is capital A small a, and small a respectively, the frequency of each genotype is frequency of recessive homozygotes will be number of recessive homozygotes in individuals in a population divided by the total number of individuals. Same is the case with the heterozygotes and the dominant homozygotes. The sum of all the genotypic frequencies always equals to 1. This is just like in case of percentage, the sum of all the percentage comes out to be 100. The sum of all the frequencies always comes out to be 1. The allelic frequency, as again the name suggests, is the frequency of a particular allele within the population. There are two different methods of calculating allelic frequencies. Either it can be calculated from the number of genotypes, or it can also be calculated from the genotypic frequencies. So, if you have the number of genotypes within a population, you can calculate allelic frequency by using this formula. The number of copies of the allele divided by the total number of copies of all the alleles at that particular locus. For example, let's assume a locus with any two alleles, that is capital A and small a here. So the frequency of the capital A allele, which is being represented by P here, is equal to twice the number of dominant homozygotes plus the number of heterozygotes divided by twice the number of individuals. Now why we are doing this twice here is we are taking twice the number of individuals because each individual carries two alleles for that particular locus. So the total number of alleles can be calculated by doubling the number of individuals. Same is the case here when we are taking dominant homozygotes. So the allele, that is capital A, can be calculated by doubling the number of individuals having the homozygotes dominant condition. So capital A, since it's coming twice, we take the total number as twice the number of homozygous dominant homozygotes. Now calculating the frequency of recessive allele, that is small a, same formula can be used, that is twice the number of recessive homozygotes plus the number of heterozygotes divided by twice the number of total individuals, that means total number of alleles. Here the number of homozygotes that are dominant in condition, the number of heterozygotes and the number of recessive homozygotes is being represented here. The alternative way of calculating allelic frequency by using the genotypic frequencies is summarized here. To do so, we add the frequency of the homozygote for each allele to half the frequency of the heterozygote, because half of the heterozygotes allele are of each type. So here we take the frequency of homozygotes plus we take half the frequency of heterozygotes for each type of allele frequency calculation, as being represented here with P and Q. Now once we have this idea about allelic frequency and the genotypic frequencies, now let us try to understand the Hardyweinberg law. Hardyweinberg law was formulated independently by two people, that is Godfrey H. Hardy and Wilhelm Weinberg, in the year 1908. The Hardyweinberg law is named differently, though the law is actually the same. It is also called as Hardyweinberg equilibrium at times, the Hardyweinberg model, Hardyweinberg theorem, or the Hardyweinberg principle. This law is actually a mathematical model that evaluates the effect of reproduction on the genotypic and the allelic frequency of a population, and thus on the evolution, because when the frequency of genotype or allele changes, it leads to evolution. If there is no change in the allelic frequency or genotypic frequency, that means there is no evolution. So this law is actually providing a mathematical model to evaluate whether the reproduction in the population is going to affect the allelic frequencies or genotypic frequency, and in turn whether it is going to cause evolution or not. It says that if certain assumptions are met in a population, then the allelic and genotypic frequencies are not altered by reproduction alone, and hence there is no evolution taking place. Now, what do we mean by these assumptions? There are certain assumptions, as we just mentioned, which are: the population is infinitely large, which in practical terms means that the population is large enough that sampling errors and other random effects are negligible. So the first assumption it requires to be followed is large population size. Second assumption is individuals in the population mate randomly. So it should follow the random mating. Third is the individuals of all genotypes have equal rates of survival and equal reproductive success. That means there is no natural selection being operated in that particular population. There is no favoritism, there is no selection being operated. Then there should be no alleles created or converted from one allele to another one. That means there should not be a case of mutation. Lastly, the individuals should not move into or out of the population. So there should not be migration, there should not be the phenomena of gene flow. So these are some of major conditions which act as assumptions so that the Hardyweinberg law can operate on a particular population. So putting it all together, if a common assumption says that a population is large, randomly mating, and not affected by mutation, migration, genetic drift, or natural selection, then two predictions can be made. The first prediction says the allelic frequencies of a population do not change. And secondly, the genotypic frequencies stabilize. That means they will not change after one generation. Now, what do we mean by this? It will be more clear when we move ahead. The Hardyweinberg law indicates that when the assumptions are met, the reproduction alone does not alter allelic or genotypic frequencies in a population, and hence no evolution takes place. Other processes such as natural selection, mutation, migration, and chances, that is the drift or the genetic drift in population, are required for population to evolve. If these phenomena are not operating as forces of evolution, reproduction alone cannot cause evolution or change in the frequencies of allele or genotype. Now, before we explain it more and try to understand it mathematically, let us elaborate more about three particular assumptions that we talked about. The first assumption we talked about was the population should be large. But what size of population is large? How to define a population as large enough so that Hardyweinberg law can operate? Theoretically, the Hardyweinberg law requires that a population be infinitely large in size, but this requirement is obviously unrealistic. In practice, a population need only be large enough that chance deviations from the expected ratios do not cause any significant change in the allelic frequencies. So it is just for a practice. Otherwise, theoretically, it is impossible to have a infinitely large population. Second exemption says that individuals in the population mate randomly. But what do we mean by this random mating? It simply means that each genotype mates in proportion to its frequencies. For example, suppose that there are three genotypes which are present in a population in the following proportions. The dominant homozygotes is 0.5 frequency, heterozygotes having the frequency of 0.4, recessive homozygotes have a frequency of 0.1. With the random mating, the frequency of mating between two capital A, that is dominant homozygotes, will be equal to the multiplication of their frequencies, that is 0.5 multiplied by 0.5 coming out to be 0.25. Whereas the frequency of mating between two recessive homozygotes, that is small a multiplied by small a, will be only 0.1 multiplied by 0.1, that is 0.01. So if the genotypes mate in proportion to its frequencies, the population is said to be called as the randomly mating population. The third assumption of Hardyweinberg law is that the allelic frequencies of the population are not affected by natural selection, genetic drift, migration or mutation. But no real population mates randomly for all traits, nor is a population completely free of natural selection for all traits. Remember, the assumptions of Hardyweinberg law apply to a single locus. It does not require random mating and other absence of selection, migration or mutation for all traits. It requires these conditions only for the locus under consideration. A population may be in Hardyweinberg law for one locus but not for the others. In the simplest case of a single locus with two alleles denoted as capital A and small a with the frequency as P or Q respectively, the expected genotype frequencies under random mating are P square for the capital A homozygotes, Q square for the small a homozygotes, and 2pq for the heterozygotes. And the sum of all these, that is p square plus q square plus 2pq, will be 1. In the absence of selection, mutation, genetic drift or other forces, allelic frequencies of p and q are constant between generations. So they are not going to change, or in other words, equilibrium is reached. Here you can see the Hardyweinberg proportions for two alleles. The horizontal axis here shows the two allele frequencies P and Q, and the vertical axis shows the expected genotypic frequencies. Each line shows one of the three possible genotypes. The frequency of the heterozygote, that is this one, is greatest when allelic frequencies are equal, that is 0.5. So the heterozygotic frequencies is greatest when the allelic frequencies are equal, that is 0.5 for each P and Q. When the frequency of one allele is high, either capital A or small a, most of the individuals are homozygotes. Also, this is another way of understanding this Hardyweinberg law. Here, the length of P and Q corresponds to allele frequencies. Here, let's say this P represents 0.6 and this Q represents 0.4. So the area of rectangle represents the genotypic frequencies. So capital A is represented by 0.6 multiplied by 0.6, that is 0.36. Capital A small a, that is heterozygotes, are represented by 0.6 multiplied by 0.4, that is 0.24, and since these two are there, so 0.24 plus 0.24 comes out to be 0.48. And the Q square, that is 0.4 multiplied by 0.4, comes out to be 0.16 for recessive homozygotes. Consider a population of monoecious diploids in which a locus has two alleles A and A in the dominant or recessive forms that occur with the initial frequencies of both the alleles as P and Q. So the allelic frequencies will be like this, as we just discussed previously. The same can be represented here with the Punnett square. So this is the male, this is the female. These are the two alleles, that is dominant and recessive one, represented by P and Q. Same is the case with the female. So there can be P square, which represents the frequency of dominant homozygotes, Q square for recessive homozygotes, and PQ plus PQ, that is 2PQ for heterozygotes. And the sum of all will be 1, as the genotypic frequencies sum comes out to be 1. This is just like P plus Q equals to 1. The binomial expansion of P plus Q square will be P square plus 2PQ plus Q square will be equal to 1, which gives the same relationship. Now, what are the implications of this Hardyweinberg law? The population cannot evolve if it meets the Hardyweinberg assumptions. because there is no change in the frequencies. Reproduction alone will not bring about evolution. Other processes or the forces of evolution must operate, are required for the population to evolve. When a population is in Hardyweinberg equilibrium, the genotypic frequencies are determined by the allelic frequencies. A single generation of random mating produces equilibrium frequencies of p square plus 2pq and q square. Now, let us see whether the Hardyweinberg law operates in the other conditions, that is the extension, such as for the loci with multiple alleles and for the X-linked loci. Hardyweinberg law expectations for loci with multiple alleles. Assume an autosomal locus with 3 alleles. So instead of just 2 alleles which we discussed previously, now there are multiple alleles, that is X1, X2 and X3, which gives rise to 6 different genotypes as listed here. According to Hardyweinberg law, the frequencies of the genotype at equilibrium depends on the frequencies of the alleles. If the frequencies of allele X1, X2 and X3 are denoted by P, Q and R respectively, then the equilibrium genotypic frequencies will be the square of the allelic frequencies, that is P plus Q plus R whole square, which gives an expression P square plus 2PQ plus Q square plus 2PR plus 2QR plus R square, where P square represents the frequency of X1 and X1, 2PQ denotes the frequency of X1, X2 and so on, which you can see here. So the square of the allelic frequencies can also be used to calculate the expected genotypic frequencies for loci with 4 or more alleles as well. So we see that Hardyweinberg law is being operated or mathematically can be used for multiple alleles as well. Moving ahead with the X-linked loci. Males have only a single X-linked allele, and so the frequency of the allele of the male genotypes are P, that is frequency of XAY, and Q, that is the frequency of XAY. Females possess two X-linked alleles, hence frequencies are P square, that is XAXA, 2PQ XA in terms of dominant and recessive, and Q square, that is frequency of XAXA both recessive. The frequency of an X-linked recessive trait among males is Q, whereas the frequency among females is Q square. This is just because the females do have only single X chromosome, whereas the females do have two X chromosomes, and that is this difference of Q and Q square is there for the X-linked loci in the males and females. Hardyweinberg expectations for X-linked loci can be further explained that when an X-linked allele is uncommon, the trait will therefore be much frequent in males than in females. Now let's try to consider it with an example of hemophilia A. It is a clotting disorder caused by an X-linked recessive allele with a frequency Q of approximately 1 in 10,000, or the frequency is 0.0001. Now at Hardyweinberg equilibrium, this frequency will also be the frequency of the disease among the males. The frequency of the disease among females, however, is Q square, as we just saw in the previous slide. So this is the value of Q. Square of it gives this expression, which is 1 in 10 million. So hemophilia is 1,000 times as frequent in males as in males, as you can see the difference here, because in male the frequency is given by Q, so there is 0.001, whereas in case of females, the frequency is Q square, so the frequency is much less as compared to the males. Now Let us also discuss what do we mean by non-random mating. Non-random mating with respect to genotype affects the way in which alleles combine to form genotypes and alters the genotypic frequency of a population. There are two types of non-random mating. One is called as positive associative and the other one is called as negative associative. In the positive associative mating, there is a tendency for like individuals to mate. Means, for example, let's take the example of humans. They do show positive associative mating for, let's say, height. Tall people want to mate preferentially with the other tall people only. Short people mate preferentially with the short people only. Whereas in case of negative associative mating, there is a tendency for unlike individuals to mate. If people engaged in negative associative mating mate for height, the tall should like the short one and vice versa. There is something called inbreeding. Inbreeding is preferential mating between related individuals. It's actually the positive associative mating for relatedness, but it differs from other type of associative mating because it affects all genes, not just the ones that determine the trait for which the mating preference occurs. So if two individuals are mating which are related, this is the phenomenon of inbreeding and it applies to all the genes. Inbreeding causes a departure from Hardyweinberg equilibrium frequencies of p square, 2pq and the q square. More specifically, it leads to an increase in the proportion of homozygotes and a decrease in the proportion of heterozygotes in the population. That is the overall effect of inbreeding or breeding between the related individuals if carried out. In a self-fertilizing population with the frequency P and Q equals to 0.5, there is an increase in homozygous individuals with each generation, as you can see here. In the first generation, the genotypic frequencies of both the homozygotes, dominant and recessive, is 1/4 and the heterozygote is 1/2. But you can see with generation, due to inbreeding, there is a decrease in the genotypic frequency of heterozygotes and there is an increase in the genotypic frequency of homozygotes. So that is the effect of inbreeding. To calculate that, there is something called inbreeding coefficient, which is measured by or depicted by capital F. It is a measure of the probability that two alleles are identical by descent. The two copies of the same alleles in homozygous diploid individuals may be the same in terms of state. That means in terms of structure and function, they are alike but do not have a common origin. Or they can be identical by descent. That means the copies are descended from a single allele that was present in an ancestor. These inbreeding coefficient can range from 0 to 1. A value of 0 indicates that mating in a large population is random, whereas a value of 1 indicates that all alleles are identical by descent. So using these values, using this inbreeding coefficient, you can predict the level of inbreeding.